CRISPR-Cas9 allows scientists to knock out particular genes and study the effect it has on cells. While this technology is not brand new, factors like time and cost have made it difficult to scale. Now, researchers in the Cheeseman lab are harnessing a new way to tease out the function of essential human genes. They collaborated with the Blaney lab at the Whitehead Institute to realize this ambitious goal. The approach involves disrupting genes using CRISPR-Cas9, then imaging cells to capture visual differences. By using powerful computational approaches, researchers can determine the regulatory relationships among genes. The team examined hundreds of cellular attributes in over 31 million cells. They probed the function of over 5,000 genes essential to human cell lines. Already, surprising discoveries are coming out of the screening data. The team discovered that two genes, AQP7 and ATP1A1, play a previously unrecognized role in cell division. Both genes' protein products transport ions in and out of the cell, a process never before associated with mitosis. This wealth of data lives online as a resource for other scientists to do their own investigations into gene function. ALS and Huntington's disease belong to a family of conditions called repeat expansion disorders. These disorders are marked by an excessive number of repeating sequences in DNA, such as CAG in Huntington's. Researchers are steadily chipping away at how repeat expansion disorders work, but some mysteries remain. For example, researchers don't know how RNA-binding proteins end up in the cytoplasm, when they're normally found in the nucleus. Previous work from the Jain lab shows that repeat-containing DNA is transcribed into RNA fragments, which clump together and hinder cell function. Now the team has found that RNA clumps can migrate from the nucleus to the cytoplasm. They believe that clumps of RNA may sweep up RNA-binding proteins, accounting for their displacement. These findings may lead to therapies that can treat or help manage these devastating diseases. A majority of cells in the body grow old and perish, but germline cells, which eventually become eggs and sperm, can pass down information through generations indefinitely. In this way, they are immortal. But how do they do it? That is the question that Whitehead Institute member Yukiko Yamashita has long explored. Germline cells undergo asymmetric division, meaning a germline stem cell will divide into another of its kind, plus a precursor to either an egg or sperm cell. Previous work from the Yamashita lab showed that cells divide asymmetrically and segregate their genetic information non-randomly. The dividing cell determines which daughter cell receives each packet of genetic information. New research from the Yamashita lab begins to tease apart why. To continue their immortality, germline cells must maintain a high number of ribosomal DNA. These are long stretches of DNA that provide instructions and materials to make ribosomes, the cellular machines that produce proteins. The team has identified a gene which they have named Indra. This gene's protein product ensures that a greater number of ribosomal DNA ends up in the daughter cell that is destined to be part of the germline. Researchers continue to study how Indra allows the germline to stay immortal. Each of our cells has 23 pairs of chromosomes, which are structures that store our genetic material. One pair is our sex chromosomes. This pair is usually an X and Y chromosome for males, and two X chromosomes for females. Researchers have long believed that in XX females, only one X plays an active role in gene expression, while the other takes a backseat, only expressing around a quarter of its genes. But new research from the Page lab shows that the inactive X plays a more expansive role than previously thought. Researchers collected tissue samples from people born with unusual numbers of sex chromosomes. Some only had one X, while others had up to 4. In people with extra X chromosomes, a single X continues to stay active, while the others are considered inactive. But Page Lab researchers made a surprising discovery. After comparing levels of gene expression between tissue samples, it became clear that the allegedly inactive X chromosome can actually affect gene expression in the active X, dialing its expression up or down. Sex chromosomes contribute to physical characteristics that we attribute to maleness or femaleness, but they do much more. they contribute to differences in all cells of the body, with broad implications for health and disease. This finding may guide researchers towards a better understanding of how diseases manifest differently between sexes.